dah ni terus tutup lah.
Okay.
Alright. So Assalamualaikum and a very good morning to everyone. Welcome to Group 6 oral presentation FSG 671 AS201. So my name is Warazana Muhammad Hassan. I'll be chairing the session for today. With me I have Dr. Muhammad Shafiq to help me in keeping the time. Dr. Muhammad Shafiq, how are you doing? Okay.
Good so far, Alhamdulillah. Nervous.
Alright, so besides me and Dr. Shafiq as the panels, we also have Dr. Fakar Zaman, Rajah here. Dr. Fakar, okay? This morning?
Okay, Alhamdulillah. Sejuk hujan sini.
And also we also have Dr. Siti Hasna. So maybe Dr. Siti Hasna is going to enter a bit late lah. And the most important is that we are going to have our 12 lovely students as the speakers today, right? So everyone is here. So very good on that. So welcome everyone. So I really hope that you are doing fine today, right? So I just want to remind the presenters that you'll be given 8 minutes for the presentation and also 2 minutes for the Q&A. So please don't be nervous, right? Just present in your pace, but within the 8 minutes, right? So, but if you exceed, of course, Dr. Shafiq will stop you. So, and then we are going to continue with the Q&A session. Yeah.
All right.
So, what happens is that make sure that once I call your name, just get ready, open your slides, right? And please start sharing, and the time will start as you start to present, right? So is that clear, everyone?
Clear. Yes, Doctor. Yeah, Doctor.
So I think we are going to start with our first speaker. So first speaker will be our lovely Miss Farah Nadia binti Muhammad Yunus.
Hi, Assalamualaikum. Can you hear me?
Yes. So you can start to share.
Okay, I will share the screen with. Can you see my screen?
It's presenting. All right. Okay, now it's working.
Okay. So can I start now?
Yes, please, Farah.
Okay. Assalamualaikum and good morning. Greetings to all lecturers and my fellow friends. I'm Farah Nadia binti Muhammad Yunus and my supervisor is Dr. Azali binti Saleh. Today, I'm going to present a review on the effects of plant growth hormones on in vitro regeneration of medicinal plant species. Medicinal plants possess many secondary metabolites such as terpene and alkaloid that offer biological properties such as antioxidant and antibacterial. The extraction of medicinal plants will be manufactured into pharmaceutical for treat and ailments. However, due to the low germination seed rate and longer maturation periods, simultaneously with the high demand for medicinal plant extraction, it leads to dwindling of their species. Therefore, in vitro regeneration are needed to overcome this limitation. In vitro regeneration is a method where explants undergo rapid cell division and differentiation in a short time period that contribute to the mass production of medicinal plants. Few factors that influence success of in vitro regeneration are explant sources, sterilization method, type and concentration of plant growth hormones, and type of culture media. In this review, we will be focused more on plant growth hormones, which are chemical substances that synthesize naturally in the plant that stimulates further developmental response toward the plant cells. Two main plant growth hormones are auxin that induce cell division to promote elongation of stem and rooting, and cytokinin that induce differentiation of adventitious shoots to promote shoot proliferation. The aim of this review is to examine the effect of plant growth regulators on in vitro regeneration of several selected medicinal plant species, namely Eurycoma longifolia, Orthosiphon aristatus, and Rauwolfia serpentina. Few research questions from this review are, firstly, what is the aim of this review article? Does auxin and cytokinin induce root and shoot formation on in vitro regeneration of medicinal plant species? Does combination of plant growth regulators, PGRS, give positive effect on in vitro regeneration of medicinal plant species? Next, does additives aid in effectiveness when combined with various types of PGRS on in vitro regeneration of medicinal plant species? And lastly, does type of soil substrates influence success of acclimatization towards medicinal plant species? Okay. proceed to the first species, which is E. longifolia or tongkat ali. Study by Hussein et al. In 2005, shoot tip explant supplemented with BAP, kinetin and zeatin showed variation in number of shoots performed, where highest shoot proliferation, which is mean average 4.0 plus minus 0.2 shoots per explant, recorded when supplemented with 5.0 mg per liter kinetin. For root induction, 0.5 mg per liter IBA recorded the highest root induction with 90% response, where the number of roots were not mentioned in the study. For acclimatization stage, the plants that transferred into the soil containing nitrogen, phosphate and potassium recorded 70% survival rate. In another study by Al-Tahir et al. In 2020, shoot induction using cotyledonary node explants supplemented with BAP, kinetin and TDZ, 1.0 mg per liter BAP recorded the highest shoot number, which is 4.87 shoots per explant. For induction, utilizing IBA and NAA, 0.5 mg per liter IBA recorded the highest number of roots, which is 3.20 root per shoot. The plants that transplant into the soil containing GV7 media recorded 85% survival rate. Next, for O. aristatus or misai kucing, study by Jayakumar and Ramalingam in 2013, shoot induction using nodal explant supplemented with 1.0 mg per liter BAP recorded maximum shoot proliferation, which is 22.23 plus minus 0.32 shoots. However, in the combination of 1.0 mg per liter BAP, 0.5 mg per liter kinetin, together with coconut water and glutamine, it significantly enhanced the highest shoot number, which is 44.07 plus minus 0.38 shoots per explant. For induction, utilizing one IBA, IAA, and NAA, 0.5 mg per liter IBA recorded the highest root induction, which is 30.27 plus minus 0.36 roots. And plants that transplant into the combination of cocoa pith and soil recorded 100% survival rate. Another study by Rashid et al. In 2015, using nodal explant supplemented with IBA and IAA, 2 mg per liter IBA recorded the highest root induction, which is 12 plus minus 0.67 roots per shoot. And the plants that transplant into the straw soil and vermicompost recorded 85% survival rate. Lastly, for Rauwolfia serpentina, study by Khan et al. In 2018, using nodal explant supplemented with BAP, NAA, IAA and kinetin individually or in combination, 2.0 mg per liter BAP recorded 12.5 shoots per explant. However, 2.0 mg per liter BAP, 0.5 mg per liter NAA and 0.02 mg per liter IAA was the best combination for inducing maximum shoot numbers, which is 13.4 shoots per explant. For root induction, highest root induction, which is 10 root per plant, were recorded when supplemented with 0.2 mg per liter IBA, and the plants that acclimatized into the soil and compost mixture recorded 100% survival rate. Study by Malik et al. In 2012, shoot induction utilizing BAP and NAA supplemented towards nodal explant, 2.05 mg per liter BAP recorded the highest shoot number, which is 6.5 plus minus 0.15 shoot per explant. However, for root induction, in the combination of 1.0 mg per liter NAA and 1.0 mg per liter IBA recorded the highest root induction, which is 5.0 plus minus 0.11 shoot per root per explant. However, only 85% plants survived when transferred into the straw soil, vermicompost and sand combination. So, in conclusion, the best type and concentration of hormones differ depending on plant species and type of explant use. For E. longifolia, 1.0 mg per liter BAP and 0.5 mg per liter IBA showed the best shoot and root formation respectively when acclimatized into the soil containing GV7 media. For O. aristatus, combination of 1.0 mg per liter BAP, 0.5 mg per liter kinetin together with additives effective for shoot induction, while 0.5 mg per liter IBA effective for root induction when transferred into the combination of cocoa pith and soil for acclimatization stage. Meanwhile, R. serpentina, combination of hormones, which is 2.0 mg per liter BAP, 0.5 mg per liter NAA, and 0.02 mg per liter IAA effective for shoot induction, and 0.2 mg per liter IBA effective for root induction when acclimatized on soil and compost mixture. Further study on plant growth hormones combination effects toward explant and the best substrate for acclimatization need to be done whether the same response recorded in shoots and root formation towards all medicinal species. Thus, it will be helpful to improve the in vitro regeneration of medicinal plant species in the future. Here's some list of references from my review. And that's all from me. Thank you.
Okay, thank you, Farah. So we open for Q&A from the panels.
Assalamualaikum Nadia.
Waalaikumsalam,
Thanks
Dr.
for the presentation. So basically, your work is about the cell growth or
Mhm.
explant growth
Mhm.
in the presence of hormone treatment. Do you have any idea on what the possible biological pathways or signaling pathway involved in the explant growth during the exposure to the hormone treatment?
Okay. Okay. For explant, we will use any type of explant, such as shoot or root. For shoot, it will be the significant pathway. For shoot, where shoot will be used to absorb sunlight for photosynthesis purpose. Meanwhile, for root explant, it will be used to absorb nutrients such as potassium, nitrogen, and phosphorus in the soil. So it will contribute to the growth of plants.
Apart from photosynthesis, are there other pathways involved that
Mm.
support the explant growth when it is exposed to the hormones? Carbohydrate metabolism or protein metabolism, are they involved?
Okay. Yes, it involved when it used for growth, and then for what's it called? Energy, which is ATP, and then for cell division.
Okay, thank you.
Thank you, Doctor.
Farah,
Yes.
so from your study,
Mm-hmm.
based on the ingredients that you have just now,
Mm-hmm.
which ingredients that you think that might that can give you the optimum results in this in your
Okay.
study?
For every species or one only?
I mean, which one is the best?
Okay. Um, okay, let me show my slide first. So, study for Mycosporine or Aristatus, the highest shoot proliferation, which is 44.07 plus minus 0.38 shoots per explant, recorded when the plant growth hormone supplement together with additive containing or it can be substituted with plant growth hormone because it provides, such as glutamine, it is an amino acid that provide available nitrogen sources that important for plant. Meanwhile, coconut water, it contain growth hormones. So the coconut water have same function as plant growth regulators.
So what you mean is that the additions of additives is very important.
Yes, it will for the sorry the
Development, the growth level.
yes the development of shoot and root induction.
Right. Thank you.
Okay.
Any other question from panels?
I think no.
Okay. So thank you, Farah.
Okay, thank you all the lecturers and my friends. Thank you.
Okay. Well done, yeah.
Um,
So can we move to the second presenter, Nazrin? Dr. Nazrin Safri?
All right. Assalamualaikum. Can you hear me?
Yes, yes.
Yes, Nazrin.
All right.
Yes. Okay.
Wait. Okay. So,
Nazrin, relax Nazrin. Senyum sikit.
Yes,
Alright.
relax. You don't need to worry that much. Just share your slides and you can start, eh?
all right. Can you see my slide?
All right, presenting, please. Slideshow, please, Nazrin. Perfect.
Can I start now?
Yes, Nazrin, please.
Hi, Assalamualaikum, and a very good morning. I bid to all the panels and my friends. I am Shanazrin bin Safri, and today I will be presenting to you my final year project, which is article review with the title Physiological Impact of Diabetes Mellitus on Bone Health. And this review has been supervised by Dr. Nodiana binti Samsul Rizal. Without further ado, let's go to the next slide. So here is the outline for my VIVA. All right, next. I will start the introduction by giving the definition. Diabetes mellitus is a chronic disease characterized by hyperglycemia resulting from the islet beta cell dysfunction and insulin resistance. It has two types, type 1 and type 2. Recently, there has been increasing evidence suggesting that diabetes mellitus also adversely affect the skeletal system. This is due to some alteration in the molecular and the cellular traits in the bone tissue. Despite extensive study on the relationship between diabetes and bone, the exact mechanism by which diabetes mellitus causes dendrimal bone effect are still elusive and poorly studied, and the diabetic bone remain as a serious health problem. So for my research question, the first one is, what is the potential biochemical routes by which diabetes mellitus affect the bone health? Is diabetes skeletal fragility associated with the microvascular complication in bone? Is type 1 and type 2 diabetes mellitus share the same mechanism on fracture rates? Does osteoblast and osteoclast activity important in bone regulation? And lastly, is there any other effective treatment regimen to bone health for diabetic mellitus patients? All of these questions will be answered in my presentation. Now move on to the objective of this review. My first objective is to highlight the potential biochemical routes by which diabetes mellitus affect the bone health, specifically on the bone fracture rates and osteoporosis. This review also discussed the role of bone cell in maintaining the balance of bone metabolism between bone formation and the bone resorption. Now let's go to the next section, which is my main body. The pathophysiology of the diabetes mellitus are closely associated with the pancreas. For type 1, the pancreas cannot make insulin or called insulin insufficient, while for type 2, it relates with the insulin resistant. The prevalence of chronic hyperglycemia from both types has been linked to the detrimental alteration in the bone structure, where it can disrupt the osteoblast and osteoclast activity. It is very important to note that osteoblast is the cell that is responsible for secreting a hormone called osteoclastin. Osteoclastin acts on the beta cell to trigger the secretion of insulin. However, damage in the beta cell eventually suppresses the production of the osteoclastin hormone, which causes some dysregulation of insulin, which later affect the both osteoblast and osteoclast activity. Then it will lead to reduce in poor bone quality and reduce the bone strength. Now I'll be explain the effect of diabetes on the bone. In bone cell, the structure called osteoblast is the cell that develop bone and involved in bone formation, while osteoclast is a specialized cell that involved in bone reabsorption, where it will absorb and remove bone. Both of this activity is very important, especially in bone remodeling process, as it will provide and maintain the bone strength. Osteoclasts maintain the balance of bone metabolism by cooperating with the osteoblast. However, the prevalence of hyperglycemia and diabetes mellitus lead to the excessive activity of osteoclasts and cause imbalance between the osteoclast and osteoblast level, and can lead to the increase in bone mineral density, BMD. Increased BMD has been associated with a decrease in overall bone turnover. Low bone turnover and increased advanced glycogen and product AGA from bone material properties has been linked to the pathophysiology of the diabetes mellitus related to the bone loss, potentially through the osteoblast apoptosis. This will increase the fragility of bone and induce the prevalence of bone fractures and osteoporosis. Diabetic osteoporosis induced by diabetic mellitus and can be characterized by a merger of altered bone quality and reduced bone mass, increased osteoclast activity, decreased osteoblast activity, increased adipogenic differentiation of mesenchymal multipotential stem cell, MSC, and increased fracture risk are all part of its pathogenesis. Now we'll move on to the discussion session. In here, I will explain the physiological impact of diabetes mellitus on the bone health. Diabetes mellitus alter the interaction between osteoblast, adipocyte, MSC, and the marrow environment. Hyperglycemia directly affects the gene expression related to the osteoblast activity, and it also affects bone metabolism indirectly by interfering with the parathyroid hormone, PTH, and vitamin D system. Increased adipocyte production fits the chronic inflammation cycle by creating reactive oxygen species, ROS, and inflammatory cytokinins, which can cause osteoblast apoptosis. The aggregation of this pathophysiology will contribute to reduction in the bone turnover and can reduce the bone quality. In this slide, I will show the mechanism by which diabetes mellitus can lead to the increase of fracture. The underlying mechanism of bone disease for type 1 and type 2 may be different due to different disease mechanism, and they are influenced by many environmental factors, including circulating hormone, different level of bone mass, oxidative stress, and level of glycogen end product. Let's discuss this feature one by one. First, hyperglycemia. It can lead to the many microvascular complications, and this may affect the bone health. Several other papers, such as in Standard Linde 2018 study, has reported that peripheral neuropathy and retinopathy are associated with the fracture rates in type 2 and also for type 1. Evidence also exists to suggest that potentially anti-angiogenic conditions, such as impaired vascular, are predominant in diabetic bone. Next, for hyperlipidemia, it will increase the phosphoinositide 3 kinase PI3K and protein kinase B PKB, which are very important in the cell metabolism and increase. Increase in PKB and PI3K will increase the activation of periform proliferator activated receptor Y, which later can increase the risk of fracture. For hyperinsulinemia and obesity, both of them has been shown to have high level of BMD, bone mineral density. Increase in BMD will cause reduction in the bone turnover and eventually contribute to risk of fracture. As a conclusion, a diabetic environment is expected to predominantly alter biochemical aspect on the bone by decreasing its organic composition and bone material strength. This occurs either directly through altered cross-link formation or indirectly through changes of cellular activity in osteoblasts, osteoclasts, and bone progenitor cell. Alteration of this factor will contribute to the fracture risk and diabetic osteoporosis. Further study, as a recommendation, further study is warranted to study the relationship between diabetes and bone, specifically on bone mineral density and other possible confounding factors such as age, sex, duration of diabetes, fracture site, and changes in bone architecture. Further study is also demanded to compare the effectiveness of treatment regimen provided on the diabetes mellitus patient by using anti-inflammatory treatment approaches such as uses anti-osteoporotic drugs. So attached here all my reference related on this review. All right, that's all from me. Thank you for lending me your ears. And now I will open for question and answer session.
Thank you, Nazrin. So we open for Q&A.
All right, Nazrin, very great presentation. Okay, Nazrin, I just like to get your opinion, point of view from your reading on several articles. So how many articles that you read for this particular report?
Thank you. For many reports is about more than 15 articles found. And I focus on different parts such as there's in bone cell in bone itself, it has bone metabolism and bone material properties, bone turnover, and also other confounding factor. So I combine all this to create a better understanding in the physiological impact, how the bone cell involved in the diabetes mellitus and can lead to the fractures rate and osteoporosis.
All right, Nazrin. Okay, now you're about to graduate from medical courses, degrees. Okay, now from your point of view, can you suggest from the various pathway that you mentioned involved in the progressions of diabetes mellitus, right, from the bone cells, which pathway that you need to tap first in order to halt the the progressions of the inflammation from here?
Right.
Any idea?
All right, thank you. That's a very good question. For me, for my personal view, I think the most contribution for the fracture risk is from the hyperglycemia, because hyperglycemia can disrupt the pancreas, where it can damage the beta islet cell, and this will cause some dysregulation in the insulin signaling pathway, and then later can lead to bone cell where it can cause imbalance between the osteoclast and osteoblast activity. And when this happens, it will cause the bone formation and bone resorption to be altered, which lead to alteration in the bone remodeling process. And hypoglycemia is most contributing factor because it can lead to many other risks of disease, such as it can cause by microvascular complication, such as in neuropathy, retinopathy, and which can, all of this can cause fall, risk of fall, and diminish in bone fracture healing. Yeah.
Thank you, Nazir. Very good thought.
All right.
Okay, so Nazrin,
All right.
in the recommendation just now, you did mention on anti-osteoporotic drugs, yeah?
Yes.
So any anti-osteoporotic drugs in your mind?
All right. During my research, I found a few called alidronate, risedronate, and teriparatide. But due to some limitation and less data on this osteoporotic drug, so further study is demanded to study, because we already know that some of the anti-diabetic medication provide beneficial impact on bone, and some provide detrimental effect on bone. So by using this osteoporotic drug, we can somehow improve future treatment, especially on fracture site and osteoporosis patient.
Okay, the one that you just mentioned, are they the frontline drugs that have been used today?
Yes, from my research is the three that I said just now, which are alendronate, risedronate, and teriparatide.
Okay, thank you.
All right.
So any more questions from the panels?
No.
Nazrin,
Yes. Yes.
you have shown that many common pathways, signaling pathways are involved in diabetic pathogenesis. So is it possible to use the existing inhibitors to treat the diabetes apart from the common one for diabetics, like kinase inhibitor, etc., to modulate the mechanism in order to control the diabetes? Is it possible?
I think it's possible, because when we talk about the physiology, it somehow relate with each other. When there's some alteration in this part, they will lead to some alteration in this part. So if that other factor that lead to some disease in our body, it can cause us to the osteoporosis and fracture rates.
Okay, thank you.
Okay, very good. But I do think that you need to find the mediators, yeah?
All right.
Okay. Thank you, Nazrin. Very good. Yeah.
All right. Thank you.
So can we have our third presenter, Miss Nurhamiza Nurbazlan?
Yes.
Miza, are you ready?
Wait.
Share your slides and can
Okay.
start,
Can you see my slide?
Please. Yes. Slideshow, please.
Okay.
All right. Perfect.
Okay. So can I start now?
Yes,
Yes, please.
Okay. Assalamualaikum and a very good morning to all panels and to all other candidates. My name is Nurhamiza binti Nurbazlan, and today I would like to present my final year project, and the title of my FYP is In-Silico Identification of Corynebacterium, Pseudotuberculosis, Transmembrane Protein using Transmembrane Hidden Markov Model, or also known as TMHMM. So next. So this is my background of study, and the bacteria in my research is called the Corynebacterium pseudotuberculosis. It is a gram-positive, rod-shaped, and non-motile bacteria. This bacteria had caused a chronic disease, both superficial and visceral form. For instance, a swollen lymph nodes and abscess formation that affect the sheep and goats. Okay, and the disease is called the caseus lymphadenitis, or also called as CLA. In my research, the method that I use is In-Silico method. This method is known to predict using the computational approach. The transmembrane protein is essential protein the transmembrane protein is a essential component of cell membrane and a key element important for its function. So the problem statement, the CLA disease caused by the Corynebacterium pseudotuberculosis had severely affected the goats and sheep industry worldwide. While many works highlight the CLA disease epidemiology and pathophysiology, little is known about the transmembrane proteins in Corynebacterium pseudotuberculosis. There is still no published report addressing the presence and role of transmembrane protein in Corynebacterium pseudotuberculosis. The objective of my study is to identify the Corynebacterium pseudotuberculosis transmembrane protein using the TMHMM server. This is the method that I use. Okay, 150 uncharacterized protein sequence of Corynebacteria were obtained from the Unipart KB website. The sequence had downloaded in FASTA format and saved in a notepad and also in the Microsoft Excel. All the uncharacterized protein are then analyzed using the TMHMM server, BLAST program, and also Cellular server. The TMHMM server were used to identify the transmembrane protein, while the BLAST program were used to compare the amino acid of various proteins or nucleotide nucleic acid sequence. Lastly, is the Cellular website to identify the subcellular localization of the protein sequence. All the data are then summarized into table and pie chart. So these are the results that I obtained. So in the biological pathway, the unknown biological pathway of the 150 uncharacterized protein, the utmost is the unknown biological pathway, which consists of 16.7%. Next is the carbohydrate metabolism with 12% and biosynthesis with 10.7%. This agreement with Araujo 2020 state that the several genes found in pathogens genome had yet to be identified. So next, for the molecular function, the DNA binding has the highest number percentage with 31.3%, followed by the second highest is the kinase activity with 13.6%, and the third highest was the unknown molecular function with 12.9%. Based on this result, DNA binding has the highest percentage. It has corroborated with Fu 2020, demonstrating that the binding and catalysis are thought to play a crucial part in anti-infection process. So next is the subcellular localization. Based on SAR 2020, one of the most important study for determining pharmacological targets is the annotation of subcellular localization. The high-risk result that I obtained is the membrane with 48%. And next, there were 38 uncharacterized protein that contain transmembrane domain with a percentage of 55.3%. So, as you can see, this table shows some of the 83 uncharacterized protein contain the transmembrane domain. But as you can see in the table, there were 26 uncharacterized protein that I shown. So, in conclusion, based on my study, the Unipart KB database, the TMHMM server, BLAST program, and also Cellular website were used to determine the transmembrane protein in the Corynebacterium pseudotuberculosis. I successfully determine the biological pathway, molecular function, subcellular localization, and the absent and present of transmembrane protein. Okay, so the major biological pathway are the unknown biological pathway, carbohydrate metabolism and biosynthesis. Next is the major of the molecular function are the DNA binding, kinase activity, and the unknown molecular function. And the most predicted subcellular localization are the membrane, and 83 of the uncharacterized protein present with transmembrane domain. To end my presentation, this study is recommended to be further studied for the development of drugs to control the CLA disease around the world. So this is all my reference that I use in my slide. That's all for me. Thank you for your attention. Okay, thank you.
Thank you, Miza. Yeah, a very good presentation. So, Miza, in your conclusion,
Yes.
you did mention about the what just now. Can you go back to your conclusion?
Here.
Right, about the biological pathways, yeah. You did mention about the unknown biological pathway, carbohydrate metabolism.
Yes.
Any idea on this biological pathway?
The idea?
Any idea what are the biological pathways? Even though you state that you have unknown biological pathways, carbohydrate metabolism, is there any? Do you have any idea? Maybe what are they? Because you did mention on the chemotaxis here.
Yeah, because I found some research paper that I used that I saw. It state that this bacteria is yet to be identified for the biological pathway, so they just state unknown biological pathway.
So until now, there is no research done on this
Yes,
carbohydrate metabolism pathway.
it is quite small.
Not even one, is it? It's quite small.
Yeah.
Oh, the research is quite small, but
Yeah.
they did not anywhere. They did not mention about any carbohydrate metabolism or biosynthesis or
They have mentioned it about the carbohydrate metabolism, biosynthesis, and many else.
No.
But the specific one, I think I cannot find it. Yeah, this one.
Oh, any other questions from the panels? From any panels?
All right, Hamiza, a very great presentation and good finding to get
Thank you.
to provide that big knowledge, right? Okay, Hamiza, in the conclusion, you have mentioned about the further development of the drugs, right? Against CLA based on this kind of based on your finding. So, from your opinion, can you suggest any study design that can actually could be used to to develop a potential drugs candidate for CLA based on your finding here? How you want to move?
We can use the omics approach to produce the drugs for this disease.
How? What's omics drug? What's omics technology platform specifically? For example, 150 uncharacterized protein. From there, for example, right? How you want to move further from all these candidates of potential proteins?
I think with all of this, like proteins, biological pathway, molecular function, all of this data can be used the omics approach. For example, like RNA sequencing, I think, approach to develop the drugs.
Okay, any reason why specifically RNA sequencing, Hamiza?
Maybe because of based on my data, there is many about the sequences. So by using all of the data, maybe we can produce the further drugs for the disease.
All right, thank you, Amiza. Very good thought, but I hope that you can read further, because I think how you want to
Doctor.
utilize your finding further, right?
Okay, okay. Thank you, Doctor, for the suggestion.
Okay, thank you, Amiza. So
Thank
can we
you,
have the next
everyone.
presenter? Mariam Shafika Mazlan. Mariam?
Hello.
Right.
Kejap Doctor.
Oh, it's okay. Just open your slides and you can start, yeah?
Alright. Okay. Do you see my slide?
Yes.
Okay.
Yes.
Okay, I'll start my presentation.
Yes, yes, please.
Bismillahirrahmanirrahim. Assalamualaikum and a very good morning to all the panelists and also all my friends. So I'm here today is going to present to you my final year project titled Natural Plant Extract as Wood and Bamboo Preservatives Again Deteriorating Fungi. And my name is Mariam Shafika binti Mazlan, supervised by Dr. Siti Rafida binti Abdul Karim. So introduction, what is wood and what is bamboo? Wood is a sustainable multi-purpose material with the biggest carbon sequestration pool on the globe. It is highly durable. Wood species are preferred and being utilized for building and construction materials owing to their characteristics and nature. It is prone to the deterioration, which is mostly caused by wood decaying fungi. Meanwhile, bamboo is the fastest growing renewable building, natural building, grows approximately 7 to 5 to 40 cm a day. Its chemical content consists of 40% to 50% of cellulose content. Compared to coniferous and broadleaf trees, their hemicellulose content is about 14 to 25% and their lignin content is about 16 to 34%. Due to its high starch, sugar, and protein contents, it is really susceptible to biodeteriorating agents such as fungi, powder post beetles, and also termites. So the idea is that we want to use bamboo as a substitute for wood coniferous trees as they are easy to grow and also act as carbon sinks. Bamboo are easily mature as they can reach their maturity by 4 years compared to other trees which almost reach 10 to 20 years to mature. Also, with the present and fast-growing population in the country, the demand for wood and other forest products is fast increasing. Deforestation and chopping down trees to be used as construction materials and furniture has significantly impacted climate change, flooding, soil erosion, loss of habitat, and the list goes on. So this is some of the example of bamboo materials as construction. So aim of the review is to identify the inhibitory effects of plant extracts towards wood degradation, fungi, mycelial growth, and also to summarize the active components of these extracts that contribute to these antifungal properties. So the gap of the study is the standard preservation of wood using toxic materials is no longer acceptable because it has a lot of negative drawbacks. The common treatments for timber is chromated copper arsenate, CCA, arsenic, creosote, borax, which can also be used to preserve the bamboo. CCA oxidation makes it much easier to leach out and contaminate the system. Hence, it was banned in many countries and are restricted or prohibited. While the direct contact with the body through improper handling application may cause skin irritation and in the extreme, it may affect the respiratory tract when inhaled, while arsenic as a human carcinogen which can cause cancer. So there are three main types of biodegrading fungi, which are mainly of soft rot, brown rot, and white rot. Soft rot is ascomycetes, which degrades on hemicellulose, cellulose, and lignin degradation is less extensive. Next is brown rot, which is basidiomycetes, degrades on hemicellulose, cellulose, and leaving lignin-rich residue. And lastly, white rot, they have a complete lignin degradation and also degrades on hemicellulose and also cellulose. So why plant extracts? Secondary metabolites are produced by plants to defend themselves against biodegradation agents. Phenols, flavonoids, tannins, terpenoids, alcohols, alkaloids are among the major categories of natural components produced by plants. Polyphenols like tannins, terpenes, and flavonoids are contained in the wood species that are naturally durable. These compounds composition are varied depending on the type of woods, its age, which part of tree or woods are taken, season of harvesting, as well as storage condition and duration. So the first plant extract is Cinnamon camphora. Cinnamon camphora belongs to the decay-resistant species. Their extracts are known to be an antiseptic and anti-infective material. Their pharmacology uses includes bronchitis, cold condition, and diarrhea. Camphor xylem methanol extract inhibited low phylum Chabrone effectively, whereas Corylus versicolor was inhibited more by camphoraxylum in chloroform extracts, even at the same concentration were applied. It is important to select appropriate solvent in order to enhance the effectivity of natural biocide compounds. So the next study conducted by Rafida et al., the extractive from camphor have potent antifungal substances including decamphor, alpha-terpenol, eucalyptol, linalool. This compound contain alcohol which is highly volatilized. So Rafida et al. Has tested Dendrocalamus asper, which is the bulu betung treated with camphor and lemongrass. Extract fits with 10% of melamine uroformaldehyde, which is the fixation, capable to be a safe preservative of bamboo, as growth of fungi were less extensive compared to the bamboo treated with the camphor and lemongrass extract alone. So the next is coffee bean. Coffee bean. Silverskin extracts of coffee bean were shown to inhibit all the wood decaying fungi studied at the highest concentration. Silverskin extracts is a skin that were removed from the bean during roasting process. So based a study by Sinevkova et al. 2021, Norway's spruce wood protection as the 2% eco solution applied by the simple technology of long-term dipping method significantly increased the spruce wood resistant to the brown rot fungus Rhodonia placenta. Silverskin eco solution at 1% concentration inhibited G-terbium about 23% and R placenta about 7%, but it did not inhibit T versicolor, which is Rhamnosus versicolor. Silverskin extract at 3% resulted in over 16% growth inhibition to the tree fungi tested on compound wood. This is because the extractive containing chlorogenic acid, caffeic, and methyl-xanthine alkaloids. So most antifungal compounds target either the formation of or the function of ergosterol, an important component of the fungal cell membrane. The knowledge on antifungal action mechanism of plant extracts is relatively limited. A possible mechanism of action is triggering of intracellular accumulation of harmful levels of reactive oxygen species ROS, resulting in oxidative stress. So when there is a external disruption or stress, this will cause an imbalanced reactive oxygen of the species and inducing the oxidative stress. Increase of RS can lead to harmful effects as the fungal membrane can be disrupted by oxidative stress, and cell can cause cell death, and the growth of the fungi can be slowed down. So anti-oxidative response to several chemical and physical factors has been studied in bacteria, animal and plant, and yeast. However, information in filamentous fungi is scarce with most of the studies. Although the accumulation of toxic levels of ROS could be a possible mechanism of action, other mechanisms must be investigated to better comprehend the inhibitory activity of plant extracts and to develop environmentally safe products to control wood decaying fungi. So to conclude, natural extractive has offer a lot of promise in terms of food preservatives, as they have a wide range of antifungal, antimicrobial, antibacterial, and antithermitic properties. Comparison between published literatures is difficult because the treatment procedures and parameters differ from one to another. Therefore, an integrated review on this subject is important. So the recommendation is that most of the literature only reviews the inhibitory effects of the extracts towards fungi growth by screening tests. A further study is required to study the efficacy of these extracts on woods and bamboos in order to be implemented in industrial processing. Several modification approaches are needed to enhance the extract's efficacy with other preservatives in order to retain the wood quality even in the harsher environment. So these are the reference that I use. That's all from me. Thank you for the
Thank you,
question.
Mariam. So we open for Q&A.
Thank you, Mariam, for the presentation. It seems that the mixture of phytochemicals are important to preserve the wood. So based on your LR, any possible methods to identify, to quantify the phytochemicals?
So there are several methods that can be used to identify the phytochemicals. Firstly, I'm using the camphor and also the caffeine, and also the caffeine. So camphor is a volatile compound, so it can be used by using DCMS. DCMS is a method where components distribute themselves between a gas and liquid phase. A gas is flowing and the liquid phase is stationary.
In GCMS, do you need to use the standard for each phytochemical?
Also can be used by using no. I don't think it can be used for phytochemicals because there's a specific test that can be used for the phytochemicals by testing the presence the presence of the phytochemicals by using several tests such as ferric chloride test, where the where the plant extract is dissolved in distilled water, and we add several drops of 5% of ferric chloride, and
So let's say you want to identify to measure caffeine using GCMS, meaning that you don't need to use any standard. Is that what you mean? You don't need to use any standard in GCMS in order to determine caffeine in your sample?
I don't think that I have a accurate answer to answer your question, but I'll take note of that and I will include in my research.
Do you know that there is a offline database for GCMS?
I know.
Okay, so basically you can use it to identify any important phytochemicals.
Okay.
Okay, thank you.
Okay.
Okay, any more questions from the panel? Perhaps Dr. Siti Hasna, you have any questions?
Thank you very much, Mariam, for your presentation. So I want to know about lignin properties. So basically you have mentioned in here that the value for the lignin content is between 16 to 34%, right?
Mm-hmm.
Okay, so I want to know, is it considered as high in value or medium in value or lower in value? How to differentiate to know whether the value is high or low to look with looking at this range?
In my opinion, its lignin content is about medium range.
How to confirm that?
Do you have any reference for this, Mariam? Like when you said that it is
no.
I want to ask you, what are the relationship or the importance of knowing this lignin content for your antimicrobial and antifungal properties?
So by knowing the properties of the lignin content that we can by knowing the content of lignin in the wood or bamboo is crucial because we want to one possible thing that we can use is to determine the fungi that can be tested because fungi has a different type of degradation mechanism. as you can see here, is a soft rod and brown rod and white rod. If they are high lignin content in wood or bamboo, they are highly susceptible to be degraded by white rod because they have a complete lignin degradation, because white rod has a lacase enzyme that can degrade directly this wood component.
Okay, thank you very much.
All right, thank you, Mariam.
You are welcome.
Thank you very much and very good. So, can we have the next presenter, yeah, which is Nur Fatini Sajida?
Yes.
Okay, Fatini, please open the slides, and please start when you are ready. Thank you.
Did you see my slide?
Yes,
Okay.
you can start,
I
Fatini.
start now. Bismillahirrahmanirrahim. Assalamualaikum and very good morning to all the lecturers and my friends. Today, my name is Nur Fatini Sajida binti Ruzani and supervised under Dr. Wan Roziano Muhammad Hassan. Today, I will present my article review on acetylcholinesterase activity of selected plants from Lamiaceae family for possible treatment in Alzheimer's disease. When doing the article review, this is my research questions, which is what is the aim of the study, what is the gap of the study, what are the main findings found from the study, what is the conclusion of the study, and what is the suggestions of the study. Okay, first of all, relationship between Alzheimer's disease and acetylcholinesterase inhibitions. As we know that Alzheimer's disease is one of the most common neurodegenerative disorder that will cause dementia, and symptom of the Alzheimer's disease is progressively memory loss, having difficulty in daily activity, and having difficulty in making decisions. Scientifically, this neurodegenerative condition will also characterize as dysfunction and severe loss of neurons in the central nervous system. And according to Singh et al., Peter Davis, who is the scientist in 1976, found a link between the memory loss and the deficits of cholinergic in the brains of the patients that suffer Alzheimer's disease. So the objective of this article review, aim of this article review is acetylcholinesterase inhibition activity with different accessions of Lamiaceae family and to determine the relationship between bioactive compound and part of the plants that contribute to anti-acetylcholinesterase activity. Okay, for this research article, I choose the Lamiaceae family, which is one of the plant family that important in the productions of essential oil because it consists of a lot of biological properties, such as example antimicrobial and antioxidant properties. And this family also often used as a substitute for synthetic products for the therapy for the various disease. And this approach by using this family will show a similar therapeutic effect as synthetic drug with less side effect that we produce by consuming the natural medicines and can be used for the and this is 12 plant species that were selected and were reviewed for this article review. Acetylcholinesterase inhibitions activity, especially in plant extractions and precipitate inhibitions, as plant extraction is the most important part of the acetylcholinesterase inhibitions, and all plant extraction will consist some level or inhibitory level activity against of acetylcholinesterase. Thus, different were used to extract the plant species and observation about the percentage inhibition was made. And the acetylcholinesterase inhibition was expressed in the percentage of acetylcholinesterase inhibitions or IC50. And different extraction of the plant species will exhibit inhibitory actions over acetylcholinesterase. This is my summarize of the acetylcholinesterase inhibitions and the table of acetylcholinesterase inhibitions that I found for the 12 plant species that were selected. I will explain a few plant species from the 12 plant species. Okay, for Rosmarinus officinalis, the method of this species will show a higher percentage inhibition of acetylcholinesterase, which is 70% compared with the aqueous extraction, which is 12%. Both extractions use 0.1 milligram per milliliter of concentrations. And the organic solvent, this show that organic solvent extraction is more effective than water extraction for this species. Next is percentage inhibitions of Mellitus officinalis and Salvia officinalis are 70.6.9% and 60.4 plus minus 5.4% respectively. And for the Lavandula angustifolia, the methanol extraction will exhibit higher acetylcholinesterase inhibition activity, which is 34%, as compared to the aqueous extraction, which is 12%. Next is relationship between the bioactive compound and part of the plant that contribute to anti-acetylcholinesterase activity. Bioactive compound in plant species will be can be observed with total phenolic contents, and the part of the plant used for the extractions were observed to obtain the total phenolic contents, and total content were presented as galley. Also can be presented as tannic acids and pyrocatechol. This is my table and summarize for total phenolic compound for the 12 species. And I will select a few species to explain. Relationship between the bioactive compound and parts of the plant that contribute to acetylcholinesterase activity, which is Salvia thylifolia and Salvia albimaculata. I choose these two species because they use a different bioactive compound to express instead. So the Salvia thylifolia use wool plants and show the highest phenolic content in methanolic extraction, which is 129.75 plus minus 0.02 milligram tannic acids, compared with the aqueous extraction, which is 72.01 milligram tannic acids. Next is Salvia albimaculata. Use the wool plants and for the methanol and ethyl acetate extraction. Methanol extraction of this species show a lower total phenolic content, which is 52.3 plus minus 0.5 micrograms of extraction, compared with the ethyl acetate extraction, which is 56.3 plus minus 0.0 micrograms. Okay, so the conclusions is plants such as Oregano majorana, Rosmarinus officinalis, Lavandula officinalis, and Salvia officinalis show a high percentage in acetylcholinesterase inhibitions. Ethanolic, methanolic, and aqua extractions are commonly used extraction in this article review. However, petroleum and ethyl acetate also show a potential solvent in obtaining a good result in acetylcholinesterase inhibitions. The content for the bioactive compound for different plants, different part of the plant will be vary with each species, and the some species will show a high total phenolic content in leaf parts, such as Salvia officinalis, but some are not. For example, Thymus vulgaris. This strengthens the hypothesis that bioactive compound different with different part of the plant vary with each species. Okay, suggestions and recommendations for this article review. As we know that various types of extraction will give inconsistent result for acetylcholinesterase inhibitions and total phenolic contents, and it made it more difficult for comparison of this family. So the recommendation is to further the study on extraction that focus on ethanolic and methanolic solvents and standardize the procedure for total phenolic content determinations. This is my a few of my references. That's all from me. Thank you.
Okay, thank you, Fatini. but your internet is quite it's not that stable because there's lagging. Right, so I open for Q&A from the panels.
Okay, Fatini, thanks for the presentation. IC50 value, I'm interested in IC50 value. It is important for acetylcholinesterase study, antioxidant, antibacterial, etc. How do you understand about the IC50 value? What does it mean by
Okay,
low
thank you.
IC50 value, high IC50 value?
Okay, thank you for the questions. As my understanding, low IC50 value will show a good inhibitions of the enzyme inhibitions. So IC50 value were used to show enzyme inhibitions in family that was studied.
Okay, you said the lower IC50 value, the greater inhibition, right?
Yes.
So how do you relate to the affinity or binding potential between enzyme and inhibitor? So
Sorry,
does
doctor.
low IC50 value mean strong binding between inhibitor and enzyme?
Strong bind.
How do you relate the IC50 value to the affinity between enzyme and inhibitor?
Patini, can you hear that?
Oh,
Can you hear that question?
um,
How do you relate IC50 with the
sorry,
affinity,
doctor. Can you
with the
repeat?
binding of the enzymes and also the inhibitor? How do you relate
Oh,
the
I see.
IC50 and the affinity of the enzyme?
Because in any inhibition situation, there must be a binding between enzyme and inhibitor. So how can IC50 value explain about the affinity between them, between enzyme and inhibitor?
Oh, I'm sorry, but unfortunately I can't answer that questions. But I will take note on that and write on my research reports.
Okay, tak apa. Terima kasih.
Thank
Any
you,
more
doctor.
questions for other panels?
All right, Fatini, very good, presentation just now. Fatini, from your opinion, in your conclusion, right, you mentioned that there are a few plants that had showing high percentage of potential inhibitory effect against acetylcholinesterase, right? Okay, among that plant, from your readings and all that, which one you wish to choose for further study?
Oh, if in my opinion, plants that I will choose on for further study is Melissa officinalis and Salvia officinalis because they only on my review they only show the accessions on ethanol, and with only one accession they show a high percentage in acetylcholinesterase inhibitions. So if I was need to choose on plant species that need to for further study, I will choose these two species because I'm sure that they will show a good results in other accessions also.
All right, thank you, Fatini.
Thank you, doctor.
Okay, so Dr. Shafiq, is the time up or?
Yes, it's up.
Okay. So thank you, Fatini. Right, so can we go for the next presenter, Sharul Nizam Shamsul Akbar?
Yes, doctor.
Right,
Yeah.
so you can open your slides and
Yeah.
start sharing, yeah?
I'm presenting my slide. Can you see me?
Yes.
Yeah.
Okay, you may start. Thank you.
Okay. Bismillahirrahmanirrahim. Assalamualaikum and a very good morning to all the panelists and audiences. My name is Sharuni Zambi Shamsu Akbar, and today I am going to present my final year project, which is an article review titled Mycosporine-like Amino Acids Profile from Algae, a review under the supervision of Madam Rafidah binti Rasul. To begin, I would like to start with an introduction of this review. Mycosporine-like amino acids is described as natural ultraviolet UV absorbing compound and a secondary metabolite that will be synthesized and commonly found in various aquatic organisms such as algae and cyanobacteria, especially when they are exposed to prolonged and high radiations of UV. With an absorbance range from 268 to 310 nanometer, MAAs have the potential to protect these organisms due to their ability to absorb UV radiation, which are UVA and UVB, without generating free radicals. MAAs from algae have been commercialized in the industry to make sunscreen, such as a product named Heliograd 365, in which cynorine and Porifera 334 were derived from the species of red algae known as Porifera umbilicalis. There are 5 research questions that I am going to highlight here. Firstly, which types of algae produce a high variety of MAAs? What are the factors that contribute to MAAs production? What are the types of MAAs produced by different species of algae? How many MAAs are common in groups of algae? And lastly, what is the area of study that should be explored more? The problem statement or gaps of this review is which of algae produce a high quantity of MAAs and what are the precise factors that lie under the MAAs production. Besides, the commercialization of MAAs from other species of algae are still scarce. Hence, the objective of this review would be to determine which groups of algae produce a higher quantity of MAAs. Apart from highlighting the objective, the significance of this review is to provide a few databases of recent MAAs since the studies in the year 1990 that comprises of different groups of algae species in which subsequently a comparison will be made. This review will provide interest for the future development of sunscreen production from other MAAs and other species of algae as well.
Sorry, Fatini? Fatini? Please mute.
Yes, Doctor.
Fatini, please mute your mic. Thank you.
Right. Alright. Moving on to the main body of this review. The first part is the algae in terms of its physiology and its commercial application. Algae are referred to as aquatic eukaryotic organisms and undergo the photosynthesis process, which means they possess chlorophyll as their primary photosynthetic pigment. However, they do not have stems, true roots, vascular tissues, or even leaves like other terrestrial plants. They also may occur in a variety of forms and size that range from micrometers to as large as 60 meters in length. Most of them are living in aquatic habitats like freshwater, oceans, and float or actively swim in the open water of lakes. Due to that, they are exposing themselves to sun radiation. Solar radiation that can reach the Earth's surface could be divided into 3, which are infrared, photosynthetically active radiation, and ultraviolet radiation, whereby it is divided into UVA, UVB, and UVC. Next, photoprotective compounds are mycosporine-like amino acids that synthesized by algae. MAAs have high molar extinction coefficients that clarify them to be the most powerful UVA absorbers in nature and efficient against UVB, which then distribute the energy as heat. MAAs are composed either by an aminocyclohexanone or aminocyclohexanimering structure that carries nitrogen substituent. Moving on to the analysis and interpretation of this review. According to Sun et al. 2020, more than 500 species of algae that have been described to contain MAAs. This pie chart shows the recent percentage of algae species in red, green, and brown algae. It is clearly seen that the major groups to contain numerous MAAs in their composition at 85% is red algae, then followed by green algae that gives 8%, meanwhile brown algae gives 7%. Next, Table 1 shows the comparison of few MAAs between red, green, and brown algae. On the other hand, Table 2 shows some examples of types of MAAs in studied species in microalgae. From Table 1, I can conclude that cynorine, porphyria 334, pectin, and Escherichia 310 are the most numerous MAAs in red algae, whereas the top 5 most common MAAs in green and brown algae are listed as shown in this slide. The pie chart as shown in Figure 4 is extracted from Table The pie chart as shown in Figure 4 is extracted from Table 1 to give a clear visualization of algae that contain MAAs. Factors that influence the production of MAAs would be prolonged of UV radiation and adaptations to the environmental changes. From a few studied research, they stated that there is no relationship between size, morphology, habitats, and position of the shore in relation to the MAAs production in algae. Before I come to the conclusion part, I would like to share some recommendations based on my review. Firstly, research and development of a new formulation of sunscreen from other MAAs should be enhanced since they have the potential to be commercialized. Next, other than prolonged and high UV radiations, the precise factors that lie under the MAAs production are poorly studied. Perhaps future research should work on this, in which subsequently it can be used for the enhancement of the MAAs production for beauty and health products. Lastly, future research to extract MAAs derived from other species of algae is expected. To summarize, this review highlights the occurrence and comparison of MAAs from different studies across the groups of algae. Besides, this review also had shown that MAAs are most found in rhodophytes compared to other groups of algae. Furthermore, future studies need to be taken to explore MAAs from other species of algae for commercialization. In addition, cynorine, porphyria 334, pectin, and Escherichia 330 are the most common MAAs produced by rhodophytes. Lastly, factors that contribute to enhancement of MAAs are UV radiation and adaptations to environmental changes. Here is the list of references of related journal articles that I have reviewed for my review article and my slides. That brings me to the end of my presentation. I would like to thank everyone here for lending me your ears and listen to my presentation. By that, I end my presentation with Assalamualaikum and thank you.
Okay, thank you very much, Sharon Nizam. So I open for Q&A from the panels. Any questions?
All right, Sharon Nizam, very great presentation.
Thank you, Doctor.
And may I know how many articles have you read for this particular review?
Oh, for more than 20 reference, to be specific, 26 of journal articles that related to my title I have reviewed.
All right, from your readings, okay, based on your references and all that, of course you will focusing more on the reported MAA production, right?
Yeah.
And from there, do you found any limited information regarding of the MAA isolation from the algae? Any reported challenge or any reported technical issues that might arise and might need to be considered?
Okay, thank you for the questions. For so the extraction of MAAs from the species have some limited due to they have various effect for the development of the MAAs. So the limitation would be the factors and the extraction methods of the MAAs from algae.
All right, thank you, Sharon Nizam.
Okay,
Thank you, Doctor.
Sharon Nizam.
Yes, Doctor.
I not know much about MAAs, but I see that the names there's numbers for the names for your MAAs. Is there any specific reasons for the numbers?
Which one? This one?
Yes, your Porphyra 334, Escherichia 330. Is there any specific reasons for the numbers?
As far as I'm concerned, there's no relation about that. It's just a name that been given by the researchers.
Oh, no specific reason.
Uh.
Okay.
Apa, Syarul, you need to read more on that, right? Okay.
Yeah,
I'm sure
surely.
that there is a reason why.
Yeah.
I do think so, yeah.
Okay.
So any more questions? Maybe Dr. Siti Hasna, do you have any questions?
Syarul Nizam,
Yes, doctor.
thanks for the presentation. If contamination happens in the lab, so can the contamination affect the MAA production by algae?
Yeah, I do think so because for the extraction of MAAs have different ways that described by different research. One can I say that some researchers have used other acids, or it's just that the distilled water for the extraction of MAAs. So I think if there is contaminations in the isolation of MAAs, there will be a disruption or there will be a contamination occur in the MAAs isolation.
So MAA production still happens, but
Yeah.
at different weights or etc.
Yeah.
Okay, thank you.
Okay, thank you, Syarul Nizam, for a very
Thank you.
good presentation. Yeah.
Thank you all panelists.
Right. So can we move on to the next speaker, Fatima Azara?
Yes, Dr. But hold on, I'm gonna open my camera
Yes.
and slide.
Please share it and you can start in. Thank you.
Okay. Can you see my slide?
Yes.
Okay. I will start now.
Yes.
Bismillahirrahmanirrahim. Assalamualaikum and a very good morning. I bid to all panels and to my fellow friends. My name is Fatima Azhar Zainuddin. The title of my review article for today is In Vitro Culture of Bambusa vulgaris, Dendrocalamus asper, Dracaena sanderiana, using different explant type, and I am under supervision of Professor Madhya Dr. Noreza. Moving on to the introduction, according to Muhammad et al., bamboo is a multi-purpose plant and is considered the second most crucial non-timber plant in Malaysia for producing valuable goods. According to Mudoy et al., it is known to be one of the most agile growing plant, and the maximum height it can achieve is between 15 to 30 meter for over 2 to 4 months. Bambusa vulgaris, Dendrocalamus asper, and Dracaena sanderiana are among the popular bamboo species. It comes with a wide range of benefits to human, from utilizing mature culm as house building to the pulp of bamboo that is used in the production of paper, and to some of the medicinal properties that these bamboo possess. Here are some of the research questions that is related to the study. What is the objective of this study? What is the most suitable growth regulators for shoot multiplication, root induction, and callus formation? Why regeneration of bamboo species is mostly done via in vitro? Why there are insufficient findings on different explant type? And what are the advantages of regenerating bamboo species via in vitro? Moving on to the objective of this study, is to determine the most suitable explant type for in vitro micropropagation of Bambusa vulgaris, Dendrocalamus asper, and Dracaena sanderiana. As reported by Mudoy et al., in vitro micropropagation has been a method used by many researchers to produce plant on large scale in a short period of time. Bamboo is no exception to the use of in vitro micropropagation for a variety of commercially and medicinally significant plant species. The production of seeds of these bamboo species is irregular. It will take a century or even more to grow seeds for some species, and it is difficult to predict the exact time for flowering to occur. In vitro micropropagation is important for the successful mass propagation of these bamboos, as it could prevent any contaminations from occurring during the development of the plants. According to reports published by Sual et al. And Kakwe and Salehi, there are some physical and chemical factors that could influence the in vitro micropropagation. The response of explant is determined by the condition of mother plants, the size of the explant, and their position in mother plants. For chemical factors such as growth hormone could also potentially influence the result of in vitro micropropagation of bamboo species. Another example of chemical factors would be the disinfectant that is used in surface sterilization, also could impact the result of in vitro micropropagation. Surface sterilization is an aseptic technique that is very crucial and has become the major factor that could affect the success rate of micropropagation. Explant that is excised from the parent plant that has been exposed to the environment must be sterilized thoroughly to ensure there is no contamination cultured in the growth medium. Surface sterilant that is frequently used to disinfect the explant of bamboo species is mercuric acid, and the standard concentration is 0.1%. According to Malini and Ananda Kumar, an increase in concentration of mercuric acid to 0.2% resulted in high aseptic culture, but it had a negative effect on the percentage of bud break. Once all the explant are free from any contaminants, it must be cultured in a medium that will act as the base for it for the explant to grow in. The culture medium is also known as growth medium in the form of gel or liquid form. It is commonly used due to favorable effect on the growth of explant and because of the price that it comes with. And the culture media, it will be enriched with growth hormones that will facilitate shoot multiplication and root induction. Picture attached is shoot multiplication of Dendrocalamus asper in liquid MS medium that is enriched with 5 mg per liter of BAP and 40 mg per liter of adenine sulfate. Moving on to the analysis of this study. These tables show the explant source, the type of explant source used for Bambusa vulgaris, Dendrocalamus asper, and D. sanderiana. From here, we can see that the nodal segment excised from adult parent of Bambusa vulgaris, cultured in MS medium that is enriched with BAP and kinetin, gives positive result in shoot multiplication, and the same result obtained when the nodal segment with axillary buds from 1-year-old seedling, also cultured in the same combination, gives the same result as well. And the nodal segment that is cultured in MS medium supplemented with IBA will induce root. Moving on to the D. asper, we can see from the explant source, most of the explant source used for D. asper is nodal segment. There's one in particular where nodal segment will either be cultured in BAP alone, or it will be cultured in BAP with a combination of adenine sulfate, and both give results in shoot multiplication. For D. sanderiana, the nodal segment of D. sanderiana excised is cultured in 2,4-dichlorophenoacetic acid and 2,4,5-triacetic acid, will give result in callus formation. Whereas the D. sanderiana, the internodal of D. sanderiana that is also cultured in the same combination of growth regulators somehow result in low percentage of callus formation. Moving on to the discussion of the study, it is proven by Aslam et al. That BAP is more effective compared to kinetin during the development of shoots. This is further supported by Nitya et al. That claim the effect of 2 mg of kinetin on shoot multiplication was insignificant. And according to Banerjee et al., the addition of adenine sulfate in MS medium supplemented with BAP helped in increasing the number of shoots, but ineffective in enhancing the frequency of response in D. asper. As reported by Aslam et al., nodal segment with significant callus biomass also had high potential for regeneration in D. sanderiana compared to into nodal segment. The use of 2,4-dichlorophenoacetic acid and 2,4,5-triacetic acid help in the formation of callus in D. sanderiana. In conclusion, nodal segment is the most suitable explant source for Bambusa vulgaris, Dendrocalamus asper, and Dracaena sanderiana. The regeneration of bamboo species through in vitro micropropagation is very crucial in producing large-scale bamboo. Large-scale production of bamboo species will be able to conserve all the bamboo species around the world. Conservation of bamboo is vital as bamboo can store carbon more quickly rather than other fast-growing tree species. For further research on bamboo species, more emphasis should be placed on incorporating alternative explant sources to be used as explant types. Here are some of the references that I used for the slide. That is all from me. Thank you.
All right, thank you very much, Fatima, for the very good presentation. Yeah. So we open for Q&A.
Okay, thank you very much, Fatima, for your great presentation. You have mentioned about the factors for in vitro micropropagation, and you have mentioned about the physical factors and chemical factors as well. I want to know what are the examples of the physical factors that would affect the in vitro micropropagation?
From my readings of all these for over 20 plus journals, I found out that from different family of bamboos such as Bambusa vulgaris, Dracaena, and also Dendrocalamus, it has some similarity in terms of physical factors. Mainly we focus on the explant, the position of explant that is excised from the mother plant, and then the condition of mother plant, whether it's an adult plant or a younger plant, and also chemical factors, the growth hormone, growth regulator mainly, and also the disinfectant used in sterilization method.
So you would say that the physical factors would be the age of the plant itself?
The explant of
the
each.
explant. All right.
Yeah.
So in between all of these species,
Mm-hmm.
okay, that you have read so far, which one is the most effective for your purpose of your research?
I would say all three species, because it actually clears out my objective of the study, which is to determine the suitable explant type. And most explant used for all three species is actually nodal explant.
Okay, thank you very much.
Okay, Fatima. From your study just now, it's very interesting to know that you use mercuric acid in the antiseptic techniques, right? You did mention that.
Mm-hmm.
Do you happen to know how mercuric acid, the mechanisms of it, of the mercuric acid in the aseptic techniques?
In terms of decontaminate the explant, I'm not thoroughly sure, but the method in sterilizing the explant that has been freshly excised from the mother plant, the researchers start with by washing the explant under the tap water, and then it will either be submerged or soaked in mercuric acid. And for the time range between 15 to 20 minutes. If more than that, it could potentially influence some of the cell that from the nodal segment, and then it will be washed with sterilized water as well.
All right, thank you. But maybe you can read more on the mechanisms, yeah?
Yes.
Okay, thank you, Fatima. Thank you
Thank
very
you,
much for the
Dr.
presentation.
Okay.
So we move on to Nur Sarah Safia binti Ali.
Yes, doctor.
All right. Sarah, can you share your slides?
Okay, wait. Is it the slide, is it clear?
Yes. Yes, you may start it.
Is my voice clear?
Yes. You may start, yeah?
Assalamualaikum and a very good morning. My name is Nur Sarah Shafia binti Ali, and today I will present my article review, a review COVID-19 and its impact on antibiotic and antimicrobial resistance. According to CDC,
Sarah,
yes.
are you planning not to open your camera?
Oh, I'm sorry. Okay. May I start? Am I need to start back at the first slide?
No need, it's okay. No need, just resume.
Okay, the International Committee of Taxonomy, the International Committee on Taxonomy of Viruses termed the virus as Severe Acute Respiratory Syndrome Coronavirus 2, SARS-CoV-2, and the infectious disease was officially renamed as Coronavirus Disease 2019, COVID-19 by World Health Organization on the same day, 11 February 2020. According to He et al. 2021, during this COVID-19 pandemic, beside the primary infection of SARS-CoV-2, many other complications are emerging, contributing greatly to the mortality, including bacterial infection, which is a significant promoter to the mortality of COVID-19 patients. According to Rodriguez et al. 2020, as the COVID-19 cases increases, the paucity of healthcare workers happens. Screening programs in identifying colonies of resistant microorganisms cannot be made to give priority to COVID-19 diagnostic and lead to empirical antibiotic prescription. These are the research questions for this article review. How COVID-19 can affect the way antibiotics are being prescribed? Does COVID-19 pandemic accelerate the misuse of antibiotics? How antibiotics can be misused during this pandemic? What is the effect of COVID-19 pandemic to the antimicrobial resistance AMR crisis? And what are the countermeasures taken to prevent the emergence of AMR during COVID-19 pandemic? The objective of this article review are to analyze the impact of COVID-19 on antibiotics and AMR, and also to identify the countermeasures taken to control the administrations of antibiotics and the surveillance of AMR during this pandemic. Next is the bacterial infection in COVID-19 patients. Bacterial co-infections. According to Garcia et al. 2021, bacterial co-infection can be defined as laboratory confirmed evidence of bacteria at the time of positive specimen collection within the first 24 hours of admission to the hospital. As stated by Grassley et al. 2021, COVID-19 patients with bacterial co-infection were highly confronted to nosocomial infection. Next is secondary bacterial infection. According to Garcia et al. 2021, secondary bacterial infection, or SBI, can be defined as infection that is diagnosed more or equal to 48 hours after admission of COVID-19 patients to the hospital. On the report of Zhou et al. 2020, SBI together with the viral replication in the lower respiratory tract leads to severe lung disease and acute respiratory distress syndrome, ARDS, which resulted in 50% of COVID-19 deaths. Next is the antibiotic usage in COVID-19 patients. Recent study from Longford et al. 2020 showed there were 3.5% of COVID-19 patients with bacterial co-infection and 14.3% of patients with secondary bacterial infection. However, the number of patients with COVID-19 that received antibiotics was recorded as 71.9%. And according to Wu et al. 2020, the large discrepancy in the usage of antibiotics and the actual number of COVID-19 patients that are infected with bacteria is generally alarming, as we are well aware that the inappropriate use of antibiotics is one of the major factors that contribute to AMR. Now I move to the factors that contribute to AMR in the era of COVID-19. Based on Rosen et al. 2020, the increase in the number of COVID-19 patients lead to the redeployment of laboratory capacity and antimicrobial stewardship AMS team to support the COVID-19 workload, resulting in less opportunity for patients to receive optimal antimicrobial therapy. On a different note, Petit et al. 2021 stated, it is also difficult to distinguish between bacterial or viral etiologies based on the presenting symptoms, and therefore in treating confirmed or suspected cases of sepsis, it can also lead to the prescriptions of empiric antibiotics. And hospital-acquired infection, HAI, is also one of the factors contributing to the emergence of AMR during this pandemic, according to Bonanomi et al. 2021. Since up to 25% of COVID-19 patients need to admit to ICU due to ARDS, which is exposed to multi-drug resistant bacterial infection. Next is the countermeasures taken to control the administrations of antibiotic and the surveillance of AMR during this pandemic, which are, according to Kubin et al. 2021, the Antimicrobial Stewardship AMS team changed the practice to ensure they were able to adapt to the challenging without changing the AMS principle. And rapid diagnostic testing with pharmacist recommendation has been conducted to decrease the risk of mortality based on Maros et al. 2020. Next is the summary of data on several studies of COVID-19 patients with bacterial co-infection. From the studies, a total of 126 cases were identified from 6,305 patients. However, only 74 of the patients were confronted with bacterial co-infection, which brings to only 1.17% of the study population. Staphylococcus species represent the highest proportion with 32 cases that brings to 0.51%. This is the summary of studies on secondary bacterial infection, SBI, in COVID-19 patients. The study showed that 208 out of 2,290 total COVID-19 patients, which bring to 9.08%, were associated with SBI. The three major species of bacteria causing SBIs were Acinetobacter species, Pseudomonas species, and Klebsiella species, with the highest was Acinetobacter species, with a total of 85 isolates, which bring to 7.58%. All three of these bacteria belongs to a group of pathogens called as the escape pathogen, which is known to be associated with nosocomial infection that contribute to high mortality and morbidity rate, according to Leo et al. 2016. Next is the summary of studies on antibiotic used in COVID-19 patients. Based on several studies conducted in China and Spain, the number of patients that received antibiotic treatment was 815 out of 1,104 COVID-19 patients, which brings up to 73.82% of the total population studied. However, the number of patients who encountered bacterial infection was only 48, or 4.62% of the study population. The prescriptions of broad-spectrum antibiotic will further accelerate the emergence of AMR. Acceleration pressure induces the growth and proliferation of resistant strain and eliminates the non-resistant strain based on Peterson and Co. 2018. And unfortunately, in combating this COVID-19 pandemic, vast majority of the antibiotics being used are the broad-spectrum antibiotics. The suggestion and recommendation I can make for this article review is that the antimicrobial stewardship should not be neglected, since it is a part of the effort to combat against AMR. Redeployment of staff, nurses, or doctors involved in antimicrobial stewardship should be prevented. Instead, extra people with professional microbiology background and pharmacists can be encouraged to help or volunteer in handling the emergence of AMR during this pandemic. These are the reference for this presentation, and that's all. Thank you.
Okay, thank you, Sarah Sakria. That was very interesting. So I open for Q&A from the panels.
All right, Sarah,
Yes.
very interesting review. Okay, from your readings, right, can you elaborate further about the, you mentioned about the Acinetobacter, Pseudomonas, and Klebsiella is among the common prevalent pathogen for the SBI, and is considered as a group as an escape, right, pathogen, ESKP. All right, so you mentioned that this bacteria, microorganism, had a high mortality and morbidity. So how these two characteristics actually increase the pathogenicity of this particular microorganism that you mentioned just now?
Okay, this is because escape bacteria is a bacteria that have the capability in antimicrobial resistant bacteria. By means, if any antibiotic has been consumed by the host, they still can survive. This is because the mechanism of these escape bacteria are drug inactivation, which is they produce the enzyme, for example, the beta-lactamase, which this kind of bacteria will produce beta-lactamase to hydrolyze the ring of to hydrolyze the beta-lactam ring of the antibiotics. And also, they also use the modifications of the drug binding site by means the antibiotic would bind to them, so they can survive in the body of the host. And also they also can change the cell permeability, which is changes the cell permeability and reduce the intracellular drug accumulation. So that's how escape pathogens can lead to mortality and morbidity rate.
All right, thank you, Sarah.
Okay, Nusara, thanks for
Yes.
the presentation. Is there any molecular basis that explains the direct communication between bacterial species and coronavirus?
Bacteria species.
Yeah.
I'm not confirm it is a molecular level, but for the bacterial co-infection does happen due to the presence of SARS-CoV-2, the virus that could induce a more drastic immuration, which is the cytokine release syndrome. And for the secondary bacterial infection, it could happen due to the reaction of SARS-CoV-2, which very much similar to severe acute respiratory coronavirus syndrome, SARS-CoV, that enable in down regulation and differential regulation of the immune gene expression of the human monocyte to create a positive environment for the SBI to grow by promoting bacterial adhesion to the host structural cells.
You mean that the human body response also support the bacteria growth
Yes.
following the exposure to coronavirus?
Yes.
Okay. Another question is, what contributes to the improper use of antibiotics during pandemics?
Pardon, sorry, doctor.
What contributes to the improper use of antibiotics during pandemic?
It because of the number of cases of COVID-19 increases, so it leads to the healthcare workers can't cope with the situation. So redeployment of laboratory capacity happens, and the antimicrobial stewardship AMS team need to redeploy. So uh, there are less opportunity for the optimum antimicrobial therapy, and also they are difficult to distinguish either the infection that affected the COVID-19 patient, either they are from bacteria or virus. So that's the thing that leads to the misuse of antibiotics.
Thank you.
All right. Thank you, Sarah Sofia. Thank you very much for the interesting presentation. Yeah.
Thank you,
So
Doctor.
we move to the next speaker, Arisha.
Yes.
Okay, Arisha,
Okay.
you may start to share, and you may start.
Okay. Wait a
Thank you.
Can you see my screen, Doctor?
Yes.
Yes. Okay. All right. Okay, can I start now?
Yep.
Please.
All right. So, Assalamualaikum and a very good morning to the panels and everyone. So, my name is Arisha Abdullah Mas'ud under the supervision of Dr. Wan Nur Hayati Wan Hanafi. And today I will be presenting my article review entitled Modifications of Extraction Buffer for CTAP-based DNA extraction from the leaves of tropical fruits. So CTAP is widely used in DNA extraction protocols because it is a prerequisite step for molecular applications such as genetic diversity analysis and phylogenetic studies. However, tropical or subtropical fruit crops contain a higher level of plant secondary metabolites, which consist of polysaccharides, polyphenols, and other metabolites such as flavonoids, tannins, and etc. So these secondary metabolites actually cause the DNA yield and DNA quality to be low, And in my research, in my review, the research questions include the purpose of this review, the gaps of study, is there any need to make a comparison on the modified protocols, and is there any relation between the extraction buffer and its efficacy in producing a good amount of DNA yield and within the good range of DNA quality? And will this review gives any significance to the future directions in research? So altogether, I have two objectives in this review. The first one is to find the correlation between the extraction buffer, DNA quality, and DNA quantity in the modified CTAP-based protocols. And second is to make the comparison by analyzing these modified protocols to extract DNA from tropical fruit leaves. And there is a gap in my study. The gap is only the parameters absorbance ratio at 280 nanometer, and DNA yield will be taken into consideration to analyze the efficacy of modified protocols. So after analyzing past studies, it can be summed up that major modifications is done by altering the constituents of CTAP buffer, which includes CTAP, beta-mercaptoethanol, PVP, and also sodium chloride. So the first one is concentration of CTAP buffer. The concentration was increased up to 3 to 3.5% instead of the usual 1 until 2%. And this actually helps in removing the contaminants such as polysaccharides. So moving on to the beta-mercaptoethanol, it was increased up to 2%, and that resulted a high yield of DNA. And according to a study done by Pendy and Hussein in the year of 2020, in the same in the protocol, they used a low concentration of 0.1% of beta-mercaptoethanol, and that resulted a low yield of DNA with high contaminations. So this actually gives us an insight that there is a relationship between the concentration of beta-mercaptoethanol and also the DNA extract. So moving on to concentration of PVP, the highest concentration of PVP used was 3%, and this was seen in almost all of the studies being reviewed. And not only PVP, but PVP and beta-mercaptoethanol are antioxidant compounds which helps in remove polyphenols. And lastly, concentrations of salts. The high salt concentration used range in 1.4 mole until 2 mole, and in a study conducted by Hekrujam in the year of 2020, the usage of sodium chloride, preferably 1.5 to 2 mole, helps to remove high amounts of proteins and also carbohydrates. So the standard quality control in my review for absorbance ratio at the 280 nanometer is in the range of 1.8 until 2.0, which indicates good quality DNA with minimal contaminations. And for DNA yield, more than 100 microgram per gram could fulfill the needs of most molecular studies. So for my research findings, the first one is pineapple. So the Taylor and Power protocol used a relatively lower concentration, which is at 1% of CTAP, 0.1% of beta-mercaptoethanol, 0.7 mole sodium chloride with no use of PVP. And this actually is the reason why the absorbance ratio resulted below than 1.8. And the modified protocol by Doyle and Doyle used a higher concentration, and that resulted a higher absorbance ratio corresponding to the DNA yield. So the second one is Musa species, which is banana. So both modified protocols use a high concentration of CTAP, beta-mercaptoethanol, and sodium chloride. However, there is a difference between the two studies conducted, which is the PVP concentration. So Shankar et al. Actually use a higher concentration of PVP, which is at 2%, compared to Siddiq, which use 1.5% of PVP. So lastly is the Mangifera indica. So all of the studies shown a good quality range of DNA, but the DNA yield actually differed from one study to another, and this is because modified Doll and Doll by Azmat actually showed the slightest variation in DNA yield because he uses a highest concentration higher concentration of beta-mercaptoethanol at 3% and 3 mole of sodium chloride. So as a recommendation to enhance the protocols, we can use a combination of a few detergents with high concentration to minimize the plant metabolites contaminations. So as a conclusion, there is a correlation between the extraction buffer, DNA quality, and DNA yield. And it can be proven by the increase in the concentration of extraction buffer with respect to the CTAP, beta-mercaptoethanol, PVP, and sodium chloride, which gave an increase in the DNA yield and quality. So these are the references I use in my slide. And that is all for my presentation. Thank you so much for listening.
Okay, thank you very much, Arisha. That was very interesting. Yeah. So I open the Q&A to the panels.
Arisha, thanks for the presentation. What is the mechanism that underlies the successful DNA extraction by CTAC?
Can you repeat the question, Doctor?
Mechanism that explains the successful DNA extraction by CTAC.
Okay, so the efficacy of the modification of CTAC buffer is analyzed by the degree of the detergents in that buffer to remove plant secondary metabolites, and it can be seen, it can be proved using the absorbance ratio at 280 nanometer and also the quantity of DNA being extracted.
I mean, how can the CTAC chemical remove the unwanted molecules from your sample just to obtain the DNA?
Okay. So beta-mercaptoethanol will helps in breaking the outer membrane of the plant cell and in activating the nucleuses in the cell so that the contaminants can be removed from the DNA extracts. And then CTAP will form a micelle around the lipid membrane, which the CTAP will capture the lipid membrane due to its amphipathic nature, and then that will lead to the release of genomic DNA of the plants. Okay, PVP will form complexes with the plant secondary metabolites such as polysaccharides, polyphenols instead, and can help in precipitating nucleic acid. And the salt concentration, at high salt concentration, the sodium chloride can actually prevent the CTAP buffer from forming complex with the nucleic acid, and rather than forming complex with the nucleic acid, the CTAP will form complex with the contaminants.
Is CTAP surfactant?
Is surfactant?
Is it a kind of surfactant, CTAP?
In my opinion, yes, because it has a cationic properties.
Okay, thank you.
All right. Thank you, Doctor.
Okay, any more questions from the panels?
No.
Dr. Shafiq, no questions?
Well, actually, because running out of time, but then, okay, Arisha, last question. Okay, you mentioned about the mango fruits and also pisangs, right, banana. So from your opinion, is there any characteristic of this particular fruit that could actually influence the requirement for you to actually use a different recipe? For example, the concentration of CTAP and other chemical reagent to get a very good yield of genomic DNA, from your opinion and reading, please.
Okay. So in my opinion, the characteristic is actually the leaf, the type of leaves, because as we know, in matured leaves, there is a higher levels of plant secondary metabolites compared to young leaves, and tropical fruit plants is considered as a perennial plant which lives more than 2 years. So this actually, the type of leaves actually leads to the determination and the choice of recipe for CTAP buffer, in my opinion.
All right, thank you, Arisha.
Okay, thank you very much, Arisha. So we move to the next presenter, which is Nur Edina Izzati.
Can you see my face? Wait.
Not yet.
Not yet. All right.
Yeah.
Okay. Okay, I will share my slide now.
Yes, please.
Can you see my slide?
Okay, perfect.
Okay,
Mr.
I will start. Assalamualaikum warahmatullahi wabarakatuh. My name is Nur Idina Izzati binti Muhammad Zahid. So today I'm going to present about my review, which is the impact of helminth bacteria co-infection. So parasitic helminth had co-developed together with a thing from 1,000 years ago and was identified by the finding of the creatures in an old dried feces sample. Recently, concurrent infection by several types of pathogen is common. The relationship may be insignificant, damaging, or may be advantageous to the host or between the parasite. Malaysia is tackling numerous challenges of infection caused by the various disease-causing parasite. However, bacterial helminth co-infection are still under investigation in Malaysia. So the research questions that have arisen are: what is the impact of helminth and bacterial co-infection? What is the prevalence of helminth and bacterial co-infection? Did the co-infection of helminth and bacteria can affect the immune system? Can the diagnosis and clinical outcomes be affected due to bystander pathogen? And what is the research gaps and future needs in co-infection in Malaysia? So the objective of my review is to summarize an overview of concomitant infection between helminth and bacteria, including the prevalence, effect on immune system, clinical outcomes, and to identify the research gap and future research in Malaysia. So basically, what is helminth? Helminth is parasitic worm that consists of flatworm, which is trematodes and cystoid, and also roundworm, which is nematodes. So entry routes and localization of helminth in human hosts can vary between helminth, which is helminth can enter the host through mouth, penetration of larvae through the skin, or bites of intermediate host vector during blood meal. And localization of helminth can be in internal organs, lymphatic and skin tissue, and also blood vessel. So naturally, host immune response will be divided into two, which is innate immunity and adaptive immunity. Innate immunity is the first immune response that will be triggered as defensive barrier. So blood monocytes are engaged into tissue and can differentiate into classically activated macrophage or M1 macrophage and alternatively activated macrophage or M2 macrophage. M1 macrophage will promote TH1 response, which is pro-inflammatory response, which are related with infection of bacteria and viruses, while M2 macrophage will promote TH2 response, which is anti-inflammatory response, which is related with helminth infection. And it is helped by IL-4, 5, 9, 10, and 13. So many helminth carry out migratory routes before reaching their end destination, which cause tissue disruption caused by helminth. This event will release an alarmins, which is IL-33 and IL-25 to activate the M2 macrophage. M2 macrophage will help in repairing the tissue and also can cause expulsion of the host, expulsion of the helminth from the host by whip and sweep process. So helminth is known as master of immunomodulator that can release excretory or secretory ES product into their host and can manipulate the host immune response in order to survive the host in the host. So there are many helminth co-infection cases reported around the world, such as helminth bacteria, helminth virus, helminth protozoa, and helminth co-infection. There are few literatures that show prevalence of polyparasitism between helminth and helminth co-infection in Malaysia. However, no recent study have been investigated on the prevalence of helminth and bacterial co-infection in humans or animals. So this table shows the prevalence of helminth and bacterial co-infection. As we can see that this country are common with tropical disease such as helminthiasis. So there has been an increasing amount of literature review on related with immune response in helminth and bacterial co-infection. So as we can see that Helicobacter species co-infection with Schistosoma mansoni show that infected T cell correlated with the bacteria may be described to distinct sites such as the liver, whereas mansoni infection is high. This can result in protection against liver damage. However, Helicobacter species co-infection with Heliosmoides polygyrus show reduction of gastric atrophy and inflammation in the stomach caused by polarization of TH1 to TH2. It was surprising that Helicobacter species demonstrate the increase of protective effect in the infected host caused by helminth infection. So Mycobacterium tuberculosis or MTB is agent of human TB. NTB co-infection with Hymonelapsis limonita or Trichuris muris can cause reduction of macrophage phagosome ability to mature and decrease the T cell activation. This caused by anti-inflammatory response with M2 polarization. This event will lead to increasing in the NTB load. Trichuris muris will increase in IL-10 production, thus reduce the capability to combat the NTB. NTB co-infection with Ascaris lumbricoid can increase phagocyte control of NTB infection, which drive the inflammatory cytokine response within the host body system. Interestingly, this event does not lead to any clinical swelling in the sign of NTB. NTB co-infection with H. polygyrus show no significant change in mycobacterial count or lung histopathological change compared to mice infected with NTB alone. Considering with all of this finding, it shows that the effect of NTB infection with different helminth will have unique result toward the severity of NTB infection. Next is study of co-infection using mouse model on effect on macrophage also have been established. Helimosomydus polygyrus, which is nematode that commonly found in mice when co-infected with Pseudomonas aeruginosa, cause increase in Th2 immune response. However, macrophage level within the lung associated with co-infection are not changing. Co-infested host survivors will increase. Co-infested host survival chances will increase due to increase in neutrophil response in the lung. So H. polygyrus co-infection with Salmonella typhimurium show no effect on the number of macrophage in the peritoneal cavity. H. polygyrus reduce the response to pneumococcal vaccination when challenged with Streptococcus pneumoniae, and helminth-infected mice were also prone to pneumococcal pneumonia. Diagnosis and clinical outcomes also can be affected due to bystander pathogen. So bystander pathogen is pathogen that co-accidentally detected along with other pathogen in the host. Co-infection of MTB with Schistosoma manzoni show that low bacterial loads in sputum and less lung cavities compared with patients with MTB solar infection. Interestingly, this diagnosis resemble HIV positive infection in comparison with HIV negative patient. So Mycosporine bovis is not only generally infect not only in cattle but can also infect the human. So Mycobacterium bovis co-infection with Fasciola hepatica can decrease in skin thickness in cattle, thus reduce the single comparative intradermal tuberculin test effectiveness. Thus this event will lead to misdiagnosed as negative bovine tuberculosis in cattle. So moving on to the discussion, impact of interaction between helminth and bacteria co-infection can be linked with multiple factors, including immune system that lead to various consequences on the host injury and cause pivotal roles in clinical outcome due to co-infection. Moreover, Th1 and Th2 immune response will be triggered prior to co-infection of helminth and bacteria, but mainly effect on Th2 due to helminth infection. A prior of dual infection can have detrimental effect on the host, increase or no change in bacterial burden, and some may be potentially beneficial to the host. Diagnosis and clinical outcomes of tuberculosis in human and animal also affected due to co-infection with helminths. To wrap up my presentation, it is recommended that further research of different interaction of bacteria and helminth, which may have distinct variation in clinical outcomes and the effect on the immune system. And investigation on the prevalence of helminth and bacteria co-infection in Malaysia could be initiated to look into concurrent impact of bystander pathogen. So this is the list of references that I use in my presentation, and that's all from me. Thank you.
Okay, thank you very much, Regina Izati. That was a very good presentation. Yeah. So I open for Q&A for the panels.
Okay, thank you very much, Irdina. I want to know how you characterize the helminth infections.
How do I characterize the helminth infection?
Yes.
Okay. Let me move to my slide first. Okay, as we can see, entry localization and helminth localization in human hosts can vary, which can be in internal organ, lymphatic and skin tissue, and blood vessel. As far as based on my reading, let's say we pick Paragominus species or nematodes live in the liver. If there is any tissue disruption in the liver, so we know that might be a helminth infection. Or we can take an example such as Schistosoma manzoni, which is it will localize in the liver, but But in this term, Helicobacter species in gastric will be affected. But Schistosoma mansona in the liver that cause the disruption in the tissue can help will be cause disruption in the tissue.
Okay.
That's all from.
All right.
Okay. My question is another question. What are the interaction between level of IgE with health mean infection? Sorry.
Level
of IgE with the health mean infections.
IgE.
Yes.
Sorry, but I didn't read any of literatures on that because I only focus on macrophage and so on. Sorry.
Actually, health means infections are characterized by the high level of IgE. All right. Okay. That's all. Thank you.
Thank you so much, Doctor.
All right, Edina.
Yes.
In one of your slides, you did state on the innate and also adaptive immune system, yeah?
Okay.
Can we go to that slide?
Okay.
All right. So, which immune is it, innate or adaptive, that this helminth will give more effect?
Against helminth infection, right?
Yes.
Based on my reading, helminth, the host immune response that will be triggered first is innate immunity, since it's the first defensive barrier in our body. However, adaptive immunity will come next if innate immunity is not longer can fight the helminth infection and so on.
Okay. So, as what you can see here, there's both can also brings you as you have helminth infection, yeah. You did state here both anti and also pro-inflammatory cytokines, right?
Oh yes.
So in your review, yeah, so does helminth infection will increase or decrease more these both pro and also anti-inflammatory cytokines?
Based on my review, I think that helminth infection will more toward the Th2 response, even though we have bacterial infection. Because
yes, that's true. And
Th2
also,
yes.
what about the pro and also anti-inflammatory cytokines? Because right now you are stating like interleukin 10. I can see your interleukin 4, yeah, your anti-inflammatory cytokines. And
Do you
also
mean by the what happened with the pro-inflammatory?
Yes, yes.
Oh,
The
okay.
cytokines.
Since that helminth will cause tissue disruption and Th2 response will cause an anti-inflammatory, which is will stop any inflammation and heal the tissue disruption. Th1 response still will be initiated, but small in number because of it is pro-inflammatory. But what I want to conclude here is that immunology itself is complex, and I'm not very sure about the behind of the Th1 response. But what I want to say here is, Th1 response we still have Th1 response, but the research has not yet been established. In It's not yet established. Okay, that's all.
Okay, thank you, Dina. Maybe you can read more on the levels of pro and also anti-inflammatory cytokines, yeah?
Okay, thank you so much,
Thank you
Doctor.
very much, right. So we can move to the next presenter, yeah? The next speaker, Nur Farah Ain binti Abdul Razak.
Yes, Doctor.
Right.
All right, am I presenting?
Yes, please.
All right. Assalamualaikum and good morning to all panels and my fellow classmates. I am Nofara Aibeti Abduraza under supervision of Dr. Khaira Asfar bin Razul. So my presentation this morning will concern on the recent development of compostable microalgae-based bioplastics.
So each year, there are 34 million tons of plastic trash being generated by mankind, with 7% of it being recycled, and also the majority of it will be ended up in our ecosystem, which in turn will affect our daily lives. So therefore, some researchers have suggested that the production of bioplastics may be the solution to curb this issue. The term bioplastics are polymers that are derived from natural resources, such as food crops, that still exhibit the same properties as synthetic plastic, but are biodegradable. Therefore, sequencing my review, microalgae, especially on spirulina and also chlorella species, have been the hot topic or subjects as the potential raw material for bioplastic production due to their rapid growth and also extensive environmental resistance, and also they do not disrupt the food chain. Moving on, these are my research questions and gaps. My research question will be answered throughout the presentation. One thing that I wanted to highlight is the significance of this study is hopefully that it would benefit the future study or the food packaging manufacturers and other industry that uses plastic materials in terms of the potential commercialization of microalgae-based bioplastics. However, the only problem of this review is that the commercialization is still in its experimental stage, because the process of cultivating the raw material and also experimenting with blending with other additives are still, in order to produce high quality bioplastics, are still in its experimental stage, and also it's very time consuming and requires a lot of investment. Therefore, the objective of the review is for me to determine the commercial viability of both spirulina and also chlorella as the potential raw material for bioplastic production, and also to discuss the recent advancement in the cultivation system that we can use on spirulina and also chlorella in order for us to improve the biomass for larger scale production. To start, I would like to start with biopolymers, which is biopolymers that are often used to produce bio-based plastics include polyhydroxyalkanoates, polyhydroxybutyrate, and also polylactic acids. According to Sina et al. In 2020, 3 of these biopolymers are indeed biodegradable, which is the main concern for this review. Hence, with that being said, spirulina and chlorella have received the most attention and also experimentation on this bioplastic product due to its ability to extract those biopolymers mentioned earlier. So spirulina, as mentioned by Sina et al., it have the ability to adapt to harsh environments such as extreme temperature, light intensity, and also extreme pH, making it very suitable for large-scale production. To further prove my point on the extraction of biopolymers, there are being recorded that 10% dry weight of PHA can be extracted from spirulina, similarly to chlorella, which can be grown in rapidly in proper nutrition, proper light intensity, making the cultivation in industry very simple. And also, additionally, 10.6% of PHB biopolymers have been recorded to be extracted from chlorella, specifically on chlorella strichi, and also up to 60% of PLA dry weight have been recorded from chlorella with high carbohydrate content. So the most concern is that how will this microalgae be turned into bioplastics? So to briefly explain, the microalgae will first go through the cultivation system, which is the main concern for my review, which I will later on explain even in depth. Next, the microalgae will go through the harvesting steps, which followed by other pre-treatments as well, in order for us to extract the specific biopolymers that we wanted. Therefore, the biopolymers will then, according to McCannon et al. In 2013, it will be blended with other plasticizers, specific plasticizers such as glycerol and also compatibilizers, in order for researchers to improve the plasticity of the bioplastic material. Next will be on the discussion part, which I've mentioned earlier. According to Costa et al. In according to Costa et al., one of the major challenges of why commercialization of microalgae base is still in the experimental stage is that it's hard to mass cultivate it in the long run. Therefore, currently, there are two current systems that are being practiced in the industry, which are open raceway pond systems and also closed photobioreactor system. So open raceway ORP system is a cultivation system that cultivate microalgae in an open environment. It's widely known in the algal research due to its uncomplicated and straightforward structure. Hence, because of this structure, equates to lower capital expense. However, because of its open environment, the microalgae that is being cultivated here have higher contamination rate, and also the researchers are often unable to control the growth environment there because it depends heavily on the environment and the location that is being cultivated. However, PBR system is a complete opposite of the ORP that is being widely used in industrial production because the manufacturer can control the growth environment of the microalgae. So the microalgae that are often used in closed photobioreactor systems are chlorella species, but the only drawback is that the ORP system is more expensive than the PBR system are more expensive than the ORP system because of its complexity and also the operation cost to maintain the operation itself. Hence, for spirulina and to produce spirulina and also chlorella in the long run for commercialization of the bioplastic packaging, researchers have found that these two systems are not economically give the economic advantages. Therefore, they have come up with the hybrid system, which could be the future microalgae cultivation for industrial wise. So these systems incorporate, according to Ferra et al. In 2019, it incorporates both of the ORP and also PBR system mentioned earlier. Specifically by Brazil et al., they have mentioned that they will inoculate the specific microalgae in PBR system, followed by the mass cultivation in ORP system. So theoretically, it would the microalgae would have the system would have low operational costs, combining with high biomass yield. To further prove this hybrid system,
Ryan,
sorry,
you have. Could you please just wrap up the presentation?
Okay. Right. All right. So the recommended that I wanted to do is based on the bioplastic, although it's still not far from production. Further studies should optimize the ratio between the both pure microalgae and as well as the pulsatizer and also additive, and also on the hybrid system. Maybe on the future study could implement on a much larger scale than what have been done in the experiment to hypothesize whether this system is really cost effective or not into this market. So in conclusion, both my reviews did answer my objectives, with each chlorella and spirulina have the potential to be commercialized as microalgae based bioplastics, and also the hybrid system could be the promising system for to cultivate this microalgae species in the future at a larger scale. And here are my references.
Okay, thank you, Ryan. So I open the Q&A to the panels.
Assalamualaikum, Farah.
Waalaikumsalam.
Okay, thank you very much for your presentation. I want to know, it's a simple one. Okay, how much is the optimum concentration for your microalgae to be added with the plasticizer in order to produce a bioplastic that you haven't mentioned before?
Oh, about that, I'm so sorry. I already know that this question is going to come up. I did search on the optimum concentration of microalgae that needed to produce one bioplastic. However, the only thing that I found is that for Chlorella species, for example, in order to for it to have the potential the potential bioplastic that is comparable to synthetic plastic, the ratio should be four plasticizers and also one chlorella. So meaning that in my opinion, I think the biomass of the microalgae should not be as much as the plasticizers.
So you would say that the plasticizer concentration would be higher.
Yes, according to the
Why?
because plasticizers are the ones that have the consistency of the plastic only, but the chlorella are the one that contributes to the biodegradability.
So what are the differences between biodegradable and compostable?
To the extent of my knowledge, I will try my best to answer. I don't know if it's true or not. I think compostable is where the plastics are compostable in soil by other microorganisms, and the biodegradability are the measurement of the compostability of the.
It's okay. You should read more on this particular matter. But the information was good. Thank you very much, Farah.
Thank you.
Okay, any more questions? Or Dr. Shafiq, do we have more time?
We don't have. Right.
Okay, so we stop here. Thank you very much, Farahin, for the presentation. So we move to our last presenter for today, Fatin Nurul Atika.
Hello, can you hear me?
Yeah, maybe you should speak a bit louder, Fatin.
Okay.
Right. So you can start to share the slides, and you may start. Fatin?
Yes. Right. Wait a minute. Can everyone see the slide?
Yes. Slideshow, please.
Slideshow, yeah. So do you wish to open your camera?
Yes. I already opened the camera. Okay.
Yes, we can see you, but we still cannot see the slide. Am I right, Dr. Shafiq, Dr. Fakarul?
I think you had difficulties to present the slide, is it? Can you just click the slideshows? I think it's not working. We can only see your screen, not just now.
Right now, I can see the slides. Dr. Shafiq, can you see the slides?
Yes.
But it's not in the slideshow, yeah?
Not in the presentation mode.
But it's not in the presentation mode yet. Have you clicked that, Fatin?
Boleh ke, Fatin? Alright. Can you save as a PDF and you present based on the PDF? Is it possible to make it a bigger?
Can you do that, Fatin? It's okay. We can wait for
It's okay. It's okay. You like it, Fatin? Always. Okay.
Fatin, is it working?
Nampak ke?
Okay. We can see you, but alright. A bit larger.
Okay.
Would that be okay for the panels?
Yes, sure.
Okay.
Boleh.
Thank you. And
Yeah,
you
boleh.
can start, Alright.
Okay. Assalamualaikum warahmatullahi wabarakatuh, and a very good morning to all of you.
Fatim, louder please. Sorry to interrupt.
Yes. Yes, Fatin. Please make it louder, yeah?
Okay. My name is Fatin Nurul Atika binti Kamaruddin, and today I will be presenting my final year project, which is a review on the potential of Morinda citrifolia in treating diabetes mellitus. This is the outline of my presentation for today. Moving on to the introduction. So, diabetes is a serious health problem. In Malaysia, 1 in 5 adults have diabetes. As reported in the International Diabetes Federation, Malaysia is a country known to have the highest prevalence of diabetes. It has been reported that 80% of people in developing countries rely on traditional medicines to treat hyperglycemia. So the reason why I'm doing this review is because it received a very high demand, because the traditional medicine receive a very high demands, and also they are less toxic and have fewer side effects as compared to other synthetic drugs. In Morinda citrifolia plant, it has been reported that it has more than 200 of bioactive compounds, including which help to induce the therapeutic effects, including anti-diabetic. The objective of the study is that I want to review the biological properties of M. citrifolia and its potential on hyperglycemic condition. Before we enter to the main part, I want to explain about the ethnobotanical of the plant species first. So, the genus Morinda is belonging to the family of Rubiaceae, which can grow up until 6 meters tall. It consists of about 80 species, and the most commonly studied species is Morinda citrifolia. In Malaysia, it is popularly known as mengkudu, but in US, it is known as noni fruit or cheese fruit. It has been natively from South Asia and traditionally used in East Africa, Hawaiian Island, and the Caribbean. So, as I mentioned before, M. citrifolia contain properties to induce therapeutic effects such as anti-diabetic, antioxidant, and anti-inflammatory activity. The key findings that I found during my review is that the fruit have bioactive compounds such as flavonoid, terpenoid, and scopolamine, which may act as substances that lower the blood glucose level. For example, in the research done by Nayak et al., they used the fermented noni fruit juice to reduce the blood glucose level in diabetic rats. So, according to them, the flavonoids in M. citrifolia act as substance that cause another substance to be secreted, which is in this case is the insulin secretion, and lowering down the blood glucose level. Another study done by McCoy et al. showing that the aqueous extract of in another study done by also Nayak and his colleagues, they used the mengkudu fruit juice combined with the temulawak rhizome juice to observe the histology of diabetic pancreas in rats. They found that alkaloid in noni fruit are able to regenerate cell death on pancreatic beta cells. Also, other substances such as saponin and also flavonoid can protect the pancreas from free radicals. The coumarins in the temulawak rhizome juice work synergistically with the properties in the mengkudu and protect the pancreas from free radicals. So this is the one, the normal rats with normal pancreatic cells, and this is the one with the combination of the fruit juice, noni fruit juice, and also the temulawak. It shows the arrows here shows the presence of the vacuolization, which the reduce of cell death. The size of the cell death is reduced in here. We move on to the next part. This is another study done by the McCoy et al. Showing that they use the aqueous extract of M. citrifolia plant, specifically the fruit, can induce the edema formation in poor rats. So they use 200 milligram of the noni extract and given orally to the experimental rat. What they observe is that the rapid changes in the pore volume of rats who observe, which proving the effectiveness of M. citrifolia fruit extract against the inflammation. However, studies investigating a direct correlation between anti-inflammatory effects of M. citrifolia and the amelioration of diabetes are still lacking. As a conclusion, Morinda citrifolia have anti-diabetic, antioxidant, and anti-inflammatory properties to reduce hyperglycemic condition. Before I ending my presentation, I would like to give suggestions and recommendations. Further research and clinical trials are recommended to confirm the efficacy and safety of M. citrifolia and also their mechanism of action in diabetic patients. From what I read, I found one of the study that they start the preclinical trials using the noni fruit juice to diabetic patients, but the number of the participants are very low. So they suggest to increase the number of the participants to confirm the efficacy of noni fruit juice. Therefore, so that in future we can introduce the M. citrifolia products into markets as a dietary supplement for diabetic patients in future. This is all the references I use for my review. That's all from me. Thank you.
Okay, thank you Fatin Nuratika for the interesting presentation. Yeah, I open the Q&A to the panels.
Thanks, Fatin, for the presentation. If you collect this plant species from different areas, from different states, etc., in your opinion, does it affect the anti-diabetic potential?
I found another study. They use a different, the same fruit, but from different country. So what the authors found out that yes, different fruit from different country will affect the potential of the therapeutic effects, but it's not too far. I mean, it's not too different.
Okay,
What
if
is important is that the way that they extract the fruit.
if that case, I mean different localities affect the anti-diabetic potential, what is the possible cause? What makes different locations affect the anti-diabetic potential?
For now, no, I have not, I do not have the correct answer for that. But from the reading, I found that the fermentation process, instead of the normal extraction, they said that the longer the fermentation process of noni fruit juice, the better the results. Because in fermented noni fruit have a higher number of phenolic compounds as compared to normal extraction.
So extraction methods affect the phytochemicals composition.
Yes, the way that they extract. And also what I found also, different parts of the plant have different amount of biological properties.
Okay, thank you.
All right. So, Fatin,
Yes.
in your anti-inflammatory activities, you just focus on the paw of the rats. Is there any specific reason for that?
I tried to find the most close to this study, but I do not found any of them. But the researchers from the paw rat study suggest that it will be useful if we conduct the investigation specifically on inflammatory effects and the amelioration of diabetes. But there is no other study on pancreatic.
So there
for
is
now.
not much study on this lah.
Yes.
Okay. Okay, thank you. Dr. Shafiq, do we have more time for the Q&A? All right. Dr. Shafiq has difficulty in the internet. So I think
Sorry.
Dr. Shafiq, I think the time is up, right, for the Q&A?
Yeah.
Yeah, all right. So thank you very much, Fatin, for the very interesting presentation.